Hi and Assalamualaikum. I am Sufatul Farzana from Group 1. We'll be presenting about external dialyzer blood leak detector. For the introduction part, dialysis is an important treatment that is used to filter the blood of the patient with kidney failure problems. It is also known as the artificial kidney as it replicates the real function of the kidney to clean the blood. It used to filter out the waste products, toxin and the excessive fluid from the blood to allow only the clean blood to return to the body. From the blood diagram itself, we can see that the blood from the patient will undergo the hemodialysis and go to the dialyzer itself where it will meet the dialysate solution. So at this point, there will be a diffusion that occur to allow all the removal toxin from the blood to diffuse into the dialysate and the clean blood will return to the patient. One of the components of the hemodialysis is the dialyzer tube itself. The working principle of the dialyzer tube is it has different tube or passage for the blood and dialysate. There are 4 tubes overall, which is for the blood to come in, for the blood to come out, and the dialysate to go in and the dialysate to go out. And we can see that between the dialysate and the blood, they will be separated by a semi-permeable membrane, in which this membrane is the one that only allows certain size of the molecule to pass through it to allow the filtering of the blood to occur. For the problem statement, the dialyzer tends to have defects like the breaking of the membrane that separates the blood and the dialysate, which cause the blood to leak. Because before this, the membrane is the one that will restrict the red blood cell size from diffusing out into the dialysate. But when the membrane is broken down, then the red blood cell can diffuse into the dialysate. This will cause the blood leak to occur, and the part at the used dialysate will have the presence of the red blood cell, and the blood loss will happen because the red blood cell is not returning to the patient. The presence of red blood cell in the dialysate then will trigger the alarm and attract the attention of nurses to come and stop the process. So as we can see from the circuit, at the part where the dialysate moving out, there is a blood leak detector consists of the photoelectric cell. Photoelectric cell will convert the light energy into the electrical energy. So when there is no red blood cell, the high intensity of light will strike to the sensor, causing the resistance of the sensor increase, so the current does not pass through it. But when there is red blood cell, the lower intensity of light will strike to the sensor, causing the resistance in the sensor will become lower and the current pass through it and trigger the alarm. The breakage of the membrane is very hard to detect visually as it involves the internal part. It becomes more critical that there are no signs of breakage that have been showed or that can be notified. So it means that the defect of the dialyzer can only be detected when the dialysis process has started and involves the real patient of the blood. So why our project is the external dialyzer blood detector? Because we have to create an early detector that can sense the defect of the membrane before we get involved with the real blood of the patient. So for the hypothesis of this proposal, first, we will study the functionality or operating principle of LDR as when it was exposed to different intensity of light. We will also investigate the effect of blood leakage towards the concentration of liquid with the absorbance of light in LDR. Then, we will study the relationship between absorbance of light and concentration of liquid to understand how blood leak may affect the sensor. Third, we will also investigate the functionality of Arduino in the construction of sensor circuits. Fourth, we will study the diffusion process that happens between dialysate and blood through the semi-permeable membrane to understand how the movement of molecules happen.
Hi, I'm Fahmi, and I will explain the experiment 1 Beer's law experiment. The objective is to determine the relationship between the concentration of solution and absorbance of light. So, Beer's law or Lambert-Beer's law is an absorption law that tells the concentration of absorbing molecules to affect the amount of attenuation. So this experiment is to study the effect of the blood leak to the absorbance of light in the LGR or the photosensor. Next is to determine the relationship between the absorbance of light and concentration of liquid. In this experiment, absorbance of light from 6 different test tubes with different concentration, which is the mixture of copper 2 sulfate and water, will be determined by using a multiplate reader. So below is the illustration of dilute copper to sulfate with different concentration. For methodology, first we prepare a beaker with 0.4 mol copper to sulfate solutions and another beaker with distilled water, and we prepare the dilutions for all 6 test tubes according to the table 1 using the pipette, which is this is the table 1, and transfer each of the solutions into the microplates using the pipette insert the multiplates into the multiplate reader to observe the absorbance of solution in test tube 1 to 6 for a wavelength of 620 nanometer, which is we follow the color, which in our case is blue, and the graph of absorbance versus concentration of solutions were plotted using the Excel. So the expected outcome is the linear graph of absorbance versus the concentrations is obtained, which determines that the concentration of liquid is proportional to absorbance of light.
Next, experiment 2 is about light-dependent resistor. The objective is to investigate the LDR operating principle. LDR is a resistor whose resistance alters in response to variation of quantity of light hitting it. When the light intensity increases, the LDR resistance goes down. This characteristic enables us to employ LDR to build light sensing circuit. In this experiment, LDR is used to mimic an optical sensor used in the blood leak detector to detect the smallest amount of blood in clear dialysate in the dialyzer. For this experiment, have 3 parts. So first, we want to see the resistance of the LDR in the daylight, shadowy and dark. Next, we want to see the changes of the LDR resistance as the voltage adjusted. And the last one, we want to see the LDR operated in the normal circuit. For the expected outcome, we want to see the resistance of the LDR decreases as the light intensity increases, as shown in this graph.
Good day, doctor. I'm Mo Pei Ling. So now I would like to present experiment 3, reading sensor with an Arduino. The objective of this experiment is to investigate the functionality of Arduino by obtaining sensor values. Introduction: the Arduino is a board based on a microcontroller. These microcontrollers are integrated circuits IC where instructions can be recorded. Instructions are written with the programming language that can be used in the Arduino IDE environment. Basically, sensor is a device that can convert physical quantities such as temperature and light to electrical quantity. There are many different types of sensor such as light sensor, motion sensor, pressure sensor, temperature sensor, sound sensor, and others. In this experiment, we use color sensor TCS3200. This color sensor can detect a wide variety of colors based on their wavelength. The color sensor has an array of photodiodes with 4 different filters. The sensor has 16 photodiodes with red filter sensitive to red wavelength, and also green filter, blue filter, and without filter that are sensitive to different color. So moving on to the next part, how does the color sensor work with Arduino? The sensor has a current to frequency converter that converts the photodiodes readings into a square wave with a frequency that is proportional to the light intensity of the chosen color. So this frequency is then read by the Arduino here. Apparatus and materials needed is color sensor, Arduino Uno, jumping wires, and color RGB. Methodology: connect the color sensor to Arduino according to this figure below, and the connection between the color sensor and the Arduino is shown here. This experiment divided into 2 parts. Part 1 is reading the output frequency. Firstly, place a different color in front of the sensor. 2 measurements here, when the object is placed far from the sensor and when the object is close to it. Secondly, read and display the output frequency on the serial monitor. Thirdly, write down the frequency value when placing different colors in front of the sensor. Part 2, distinguish between different colors. Firstly, insert the frequency peak previously on the cord. Secondly, sensor distinguish between different colors, red, green, blue, and no filter. That is clear. This is the expected outcome. The blue frequency should be the lowest compared to the red and green frequency readings we can see in the result here. So blue is the lowest. There are 3 conditions when distinguish between different colors. When the R is the maximum value in RGB parameters, there is a red object. When G is the maximum value, there is a green object. When B is the maximum value, there is a blue object.
For the last experiment, experiment 4 is about diffusion process of semi-permeable membrane. The objective of this experiment is to study the diffusion process as in dialysis. Diffusion is a term used to describe the flow of individual molecules of a substance through a semi-permeable barrier from a region of higher concentration to a region of lower concentration. Whisking tube is used as a synthetic membrane that is selectively permeable to tiny molecules such as water and glucose, but impermeable to larger molecules such as starch and sucrose. So for this experiment, glucose and starch will be mixed in the whisking tube and left in a beaker submerged with water for about 30 minutes. After that, the iodine test and Benedict test will be performed for the fluid inside the beaker and inside the tube. For the expected outcome, we expect that when the iodine is added to the solution in which starch is present, the solution will turn blue, black or purple. Otherwise, it will remain yellow amber. And when the Benedict reagent is added to a solution in which reducing sugar is present, in this experiment is glucose, and then it is heated in the water bath, the solution will turn from blue to green to yellow, orange, red and then brick red. Otherwise, the solution will remain blue. For this experiment, we expect that iodine test will give positive result in the content of the whisking tube, but negative result for the content of the beaker, and the Benedict test will give the positive result for the both content in the whisking tube and the beaker.